In our lab, we maintain a Megalurothrips usitatus colony in a controlled environment, using insect-proof cages as a source of insects for experimental assays. We also have a predatory mite colony maintained in these rearing areas in a growth chamber, which provides mites for our predation studies. For our predation studies, we use a modified Munger cell design as arenas for our assays. To build up the arenas for predation studies, we start by preparing a leaf disc, placing it on wet cotton to keep the leaf healthy and maintain moisture. Then, we use acrylic slides, which are stacked together to form the experimental unit. In this predation study, we transfer Megalurothrips usitatus larvae into the experimental arenas, followed by the introduction of a predatory mite. We observe its feeding ability on the thrips larvae, as well as its initial behavior upon entering the arena, recording how it interacts with the prey. Additionally, we monitor whether it reproduces while feeding on this prey type. In these predation studies, we're evaluating Aureus as a predator for M. usitatus. We use modified Munger cell arenas, similar to the design mentioned earlier, but with larger acrylic slides. Then, we introduce a female Aureus, allowing us to observe how it initially interacts with the prey and to measure how much prey it consumes each day. In our lab, we also test entomopathogenic nematodes to control thrips. These nematodes are reared and multiplied in waxworms and are used in virulence studies. The primary objective of these studies is to screen commercially available biological control agents for their potential use against Megalurothrips usitatus. This serves as an initial step before testing their efficacy in more realistic environments